Hello everyone, I'm excited to gain some knowledge on one of the cutting-edge gene-modifying techniques and how it can be used to treat lethal diseases. Now let's welcome today's main player, RNAi. RNAi, also known as RNA interference, is a mechanism based on double-stranded RNA to silence gene expression. It has been widely used in gene therapies to knock down disease-related genes. What is so special about RNAi? Some of its advantages are it's a highly efficient and selective endogenous process to treat a disease. It's rapid and cost-effective and also target multiple genes simultaneously in a drug. RNAi is widely used to treat multiple genetically inherited diseases, and this video will mainly focus on RNAi's role in treating one of the most widespread neurodegenerative disease, Huntington's disease. First, let's get some background of Huntington's disease. Huntington's disease is an autosomal dominantly inherited disease, which means carrying one of the defect allele is sufficient to obtain the symptoms. The presence of 40 and more CAG triplets in exon 1 of the Huntington HTT gene has been identified as a fully penetrant trigger for the neuropathological process, which usually stretches over decades. The resultant neuronal death affects primarily GABAergic median spiny neurons in the early stage of Huntington's disease, as well as neurons in other brain regions in the later stages. The CAG expansion translated into a polyglutamine track in the N-terminus of the HTT protein, causing the mutant protein to misfold and aggregate. Therefore, clearance of the mutant protein is currently accepted as being key for Huntington's disease treatment. So how does RNAi work? It either blocks translation or causes degradation of the transcript. Two types of RNAi is usually used, respectively known as siRNA and shRNA. Although they are introduced into a cell in different ways, they merge during the formation of RISC enzymatic complex stage. siRNA, also known as short interference RNA, is introduced into a cell as double-stranded RNA, while shRNA is a synthetic RNA molecule with a short hairpin secondary structure. It's delivered on a DNA plasmid, which allows it to be continually expressed for months and even years. After the RISC complex is formed, the RNA strand separates, and the antisense guide strand directs RISC complex to degrade complementary target mRNA, such as the HTT protein. This table shows the comparison of siRNA and shRNA. In summary, siRNA has a more transient effect and usually requires repeated dosage with higher off-target effects. It's usually used for acute disease conditions. shRNA, on the other hand, can express in the cell for longer time and will cause fewer immune responses, but it's hard to control its expression level, which may lead to oversaturation of the cell with toxic level of RNAs. shRNA is therefore usually used to treat chronic diseases. We'll now look at one recent study which uses HT model mice to investigate how RNAi may be used to target the symptoms of HTT accumulation. To generate the HD rat model, wild-type rats were injected intrastriatally with a lentivirus expressing a chimeric mutant HTT fragment, which is shown to induce local formation of mutant HTT aggregates, followed by severe neuronal dysfunction at 2 months post-infection. As shown in Figure 1, AAV5 viruses carrying the miRNA expression cassettes is co-injected with LV vector, which is consisted of an 82-long glutamine chain. Each virus is injected bilaterally in the striatum of rats with their LV vector, encoding a chimeric human mutant HTT sequence. The mutant HTT protein that is expressed from both LV vectors is known to cause HT-like neuronal dysfunction. There's also a green fluorescent protein, GFP sequence in the construct to assess transduction efficiency. So what are the results? In order to evaluate the AAV5 vector distribution in the HD rat brain, immunohistochemistry against GFP on the fixed striatocortical sections is performed. For all AAV5 constructs, there's broad striatal with partial cortical GFP distribution, which is shown by the red arrows in Figure 2a. In Figure 2b, there's also approximately 1,800 times more MIHTT molecules in the AAV5 MIHTT injected rats compared with the saline control. The accumulation of mutant HTT aggregates in the brain is a hallmark of the HT neuropathological process. To validate on-target silencing efficiency of the expression products, IHC is performed using an anti-HTT antibody that binds to the mutant HTT. There are significantly fewer mutant HTT aggregates in the HD red striatum injected with the viruses compared with the saline control as shown in Figure 2D. In conclusion, RNAi has been proved to be an effective way in treating Huntington's disease. Although much progress has been made using rodent models, there are still doubts how these results can be applied to human cases. Future studies may be looking at how they can minimize the side effects and lower the dosage used. Thanks for watching this video, and I hope you have learned something from this. Also, if you know anyone with Huntington's disease around you, please do your best to help them.